Greetings, Lions. I am past District Governor Lion Nancy Mathwig from District 5M2 and have served as the host committee chair for this 2021 MD5M convention. I have also had the privilege of serving on the Minnesota Lions Vision Foundation as secretary. The Vision Foundation has been supporting Lions Gift to Sight, formerly known as the Minnesota Lions Eye Bank, since 1960, and in fact was founded for just that purpose, to help the blind and visually impaired. Today, I have the honor of introducing Dr. Joshua Ho, who will speak to us about the research that has been done during the past year. Joshua Ho graduated summa cum laude from Duke University in 2005 with a Bachelor of Science in Chemistry and minors in Mathematics and Religion. He then received his MD from Washington University School of Medicine in St. Louis in 2009. Following an internship year at Inova Fairfax Hospital under the affiliated Georgetown University Hospital, he completed an ophthalmology residency and a fellowship in cornea and external diseases at the University of Illinois Eye and Ear Infirmary, where he was awarded Fellow of the Year honors. After graduation, Dr. Ho accepted a faculty position at the University of Minnesota, where he is an assistant professor of ophthalmology with the Cornea Service. He serves as the medical director of Lions Gift to Sight and is the principal investigator of a Minnesota Lions-funded basic research lab. His current research interests include corneal limbal stem cells, as well as optimizing the quality of endothelial corneal transplants. As Dr. Ho recently stated to the Vision Foundation Board, the eye bank continues to establish itself as a premier eye bank in ocular research and in eye banking research, and is well respected in the community. They are establishing themselves as a premier research eye bank in the country and developing collaboration with the university researchers across the world. I have the honor today of introducing Lion, Dr. Joshua Ho, who is also a member of the Minneapolis Ambassadors Club. Dr. Ho.
2, 1.
Okay. Hi, my name is Dr. Ho. As you know, I'm one of the cornea faculty at the University of Minnesota. I'm also the medical director at the Lions Gift of Sight Eye Bank. I'm very grateful to be here as a speaker for the Minnesota Lions Vision Foundation, and I want to share with you some of the things that the Vision Foundation has supported and some of the sort of cool things that have come out of their support. So as medical director for the eye bank, I of course have a bias towards all the great things that are happening at the eye bank, and I just want to emphasize again that the Minnesota Lions Vision Foundation has something really amazing at the Lions Gift of Sight Eye Bank. The eye bank is one of the top research eye banks in the country, and actually in the last 6 months we've really established ourselves as the preeminent eye bank for researching COVID-related issues to donor tissue for the eyes. And basically the EBAA, the Eye Bank Association of America, and researchers in eye banking all around the world are looking at what the Lions Gift of Sight Eye Bank is doing currently. And I want to share more about that with you guys today, just so you can really sort of get excited about what's happening right now and all the good work that's being done. So today I'm just going to go through this presentation. This is just an update on all the COVID-related research that we've done, and a lot of this has been supported by the Minnesota Lions Vision Foundation. So just quick background, as you all know, COVID is basically a global pandemic caused by a virus which is called SARS-CoV-2. It was first described in Wuhan, China, and it has spread globally and has caused major public health issues. In the US alone, there have been more than 30 million infected cases and over 560,000 deaths to date. The SARS-CoV-2 virus is primarily a pneumonitis, meaning it primarily infects the lung, and that's how people tend to die from the disease. However, there is increasing evidence that the SARS-CoV-2 virus can also infect the eyes. And this has an impact on ophthalmology and also eye banking, since we transplant corneas for people who suffer from corneal scarring and other diseases. The CDC has recommended eye protection as a routine precaution for people who care for infected COVID-19 patients. They also recommend eye protection for people like ophthalmologists who are in close contact with patients The EBAA, which is the Eye Bank Association of America, has also issued very strict guidelines for screening out donors who might be infected by COVID-19 in order to rule out tissue that might be contaminated and reduce the risk that we transmit the disease through transplantation. Now, what is the current evidence for ocular infection? Well, there's clinical cases of ocular transmission. So there's anecdotal cases out of China and in the US where physicians or healthcare workers have worn full PPE, personal protective equipment, including masks, N95 masks, gown, gloves. However, they didn't wear protective eyewear and they ended up getting COVID-19 after interacting with COVID-19 patients. There has also been evidence showing that you can isolate the RNA of the COVID-19 virus from the tear film or from swabs of the conjunctiva, which is the white part of the eye. You can swab the surface of the eye and you can find COVID-19 RNA from these samples in patients who have COVID-19. Not in all patients who have COVID-19, but in a good number of patients who have COVID-19. They've also shown that if you look at all the donor corneas that come in and are screened out for COVID-19, about 20 of these corneas, you can detect the RNA of COVID-19 virus in these tissue samples. And so we do know that COVID-19 seems to be able to at least contaminate the ocular surface and corneas that we bring in from COVID-19 positive donors. So this has a major impact on eye banking, not just in the US, but globally. And in fact, COVID-19 has really limited our supply of tissue. Many donors are now being ruled out because of the possibility that they could have COVID-19. In the early days of the pandemic, we ruled out everyone who had fever and cough, and that could be a lot of potential donors because a lot of people who die from various causes can have fever and cough that's unrelated to an actual infection. And so based on EBAA reports, eye banks are actually still operating at a reduced sort of tissue volume. They have less donor tissue than they used to before, and about 81% of the tissue volume is now available compared to before COVID-19. And then European eye banks are operating at an even lower rate, about 70% of what they had before COVID-19. And this is again because we have to rule out tissue that might be infected by COVID-19. Many of these corneas do not have COVID-19. Like I said, only about 20% of the ones we rule out actually end up having the virus in the tissue. But in an abundance of precaution, we have to rule out anyone who could be so that we can catch all these contaminated tissues, and that's limiting the supply of tissue that we have for transplantation. That's making it less and less available for surgeons to use to treat people who have corneal blindness. So one of the big questions then is, well, we're doing all these screenings and we know that the tissue can be contaminated, but the tissue itself, is it actually being infected by the virus, or is it just kind of hiding on the surface of the eye like the virus can hide on the surface of a tabletop or on your hands? Is it really infecting the corneas? And if it's not really infecting the corneas, is there really a risk that patients who get transplanted a contaminated cornea will actually develop the COVID-19 disease? There's been a lot of interest in this, and obviously we're not the only group working on it, but our eye bank has become one of the leaders in this sort of research topic because of its impact on eye banking. So one of the things that determines whether the cornea or the ocular surface can be infected by COVID-19 is whether the ocular surface and the cells on the surface of the cornea actually have receptors that allow the virus to bind and then enter into the cell and set up an infection. You can think about this as a situation where you have to have certain hardware for the virus to be able to enter into the cells. It's almost like a lock and key system where the virus has the key, but the lock has to match the key, and if they match, then the virus can enter the cells. The locks that we know are important for viral entry are the ACE2 receptor and the TMPRSS2 receptor and the DC sign R receptor. ACE2 in particular is important for binding. The TMPRSS2 allows the virus to then enter the cell after the binding, and then DC sign is an alternative pathway for viral entry. So these are all important factors that allow entry of the virus into the cell. And like I said, it's kind of like trying to figure out whether the virus, which has like a screwdriver head, fits the screw on the cell. And if it doesn't fit, then you can't have viral entry. But if it fits, then you can have viral entry and infection. And so what we did was we looked at these three viral entry factors, ACE2, and we looked at it in different cells on the cornea. And we looked on the corneal epithelium, which is the clear skin that grows on the surface of the cornea. We looked at the corneal endothelium, which is the layer of cells that grow on the back of the cornea, and they're the most common cell type that we transplant in corneal transplantation. And we also looked at the limbus, which is where the stem cells that regrow the clear skin of the cornea, where those cells live. And then we also looked at the conjunctiva, which is the white part of the eye, which is where we know that RNA from virus has been harvested from before. And what we found is you can see the green stain highlights this particular receptor, and you can see that there's green staining in basically every single cell type. And this is consistent with the fact that the cornea expresses the appropriate hardware for the virus to enter into the cells. We also looked at TMPRSS2, which is again an important enzyme that mediates the entry of the virus after it's bound to the ACE2 receptor. And here it's stained in red, and you can see in the different cell types from the corneal epithelium to the conjunctiva to the corneal endothelium, you have expression of this receptor, and this allows the virus to enter into basically all the cells on the surface of the eye and on the endothelium. And so again, we've identified that the hardware does exist. And then DC sign R is an alternative pathway. In case you don't have ACE2, the virus can use alternative pathways to enter into the cells. And again, you see it light up in green in all the cell types in the cornea. And so we were one of the first groups to publish this, actually in the early days of the COVID-19 infection, showing that the hardware that is necessary for the virus to infect the surface of the eye is there. And so this is evidence that COVID-19 can be present in the eyes of patients who are infected with COVID-19, but also in donors who wish to donate their eyes after death. And here again, you can see what we did was we actually took the cells from a donor cornea. We actually cultured them out. We actually cultured them out in the laboratory, and then we wanted to test whether these cell cultures could also be infected. And because if you can infect the cell cultures, that allows you to do more studies on the virus. And we did find that ACE2, DC sign, and TMPRSS2 are all present in cell cultures that are taken from the cornea, limbus, and conjunctiva. And so, again, this is all evidence that the cells are susceptible to infection, and this was, again, one of the earliest evidences that we had that the cornea could be infected. And like I said, our eye bank was just at the cutting edge when the COVID-19 outbreak happened in defining this. So we know that the cornea has the hardware to be infected. It is susceptible, but can it actually be infected? Can these donor corneas be infected if you actually apply live virus to the cornea? So having the hardware is just sort of a theoretical thing. Yes, the key can fit into the lock, but does the key actually open the door? And so in order to figure out whether corneas could actually be infected, we wanted to do live viral infection studies. So what we wanted to do was we wanted to take these corneas and these cell cultures from these corneas and actually infect them with live virus and see if we could actually infect the tissue. And we did this in the lab. We did this in collaboration with our virology colleagues at the University of Minnesota. So luckily we have great experts from all fields of medicine here at the University of Minnesota. And we have access to a biosafety lab level 3. So you can't do research on SARS-CoV-2, the virus which causes COVID-19. You can't use the live virus unless you have a special lab that prevents it from getting out into the world. Part of the problem with Wuhan was they were using this virus and they were studying the SARS virus, the original SARS virus, and it seems like it got out because they didn't have the appropriate mechanisms in place to keep it from getting out. And so in the US, this is tightly controlled. You have to be in a biosafety lab level 3 to be able to manipulate the live virus. And there's only a couple dozen of these around the country, and we're fortunate that we have it at the University of Minnesota. So with our virology colleagues and access to these BSL level 3 safety labs, we were able to basically take cells from the cornea. We took them from the surface of the cornea, the corneal epithelium, from the conjunctiva, which is the white of the eye. We also took it from the endothelium, which is the cells on the back of the cornea. We took it from the stem cells that grow on the surface of the eye. So we took all the different cell types from the cornea. We cultured them out into petri dishes in the lab. And then we took live SARS-CoV-2 virus and we infected them. And then we tested the cultures over time to see if we could see replication of the virus in these cells and active infection. And then we did another test where we actually just took a whole donor cornea and directly infected the entire cornea itself, not just the cells that were cultured out, but the entire corneal tissue as a whole. And we did that to determine whether donor corneas could be actually infected. And so here's some of the results. This is a sample of conjunctival cells that were grown out in culture. All the blue spots are different conjunctival cells that are grown out in a petri dish. The red ones are cells that have been infected by the virus, and you can see that there's infection of the virus scattered throughout the cell cultures here, and here's the overlay where it's blue on red, and you can see where the cells have been infected. So the conjunctiva can be infected. We did this study with 2 different viral strains. At the time we had the strain from Hong Kong, which was very similar to the Wuhan strain, and then we had the strain from Washington, which was the initial outbreak that happened in the US. So we had the Hong Kong strain and the Washington strain. Now there's multiple strains around the country, and there's even concerns that there's like a more highly virulent mutated strain from Europe that's running around. There was a strain in Arizona that was thought to be quite bad that kind of happened more in the fall. But we had these 2 strains, and these are the 2 strains we tested, and you can see both strains were able to infect the conjunctival cells. Here we have the Hong Kong strain, and it is showing infection of the stem cells that grow on the surface of the eye, where the white part of the eye meets the clear part of the eye. And again, you can see the red staining of the cells that have been now infected by the live virus, again proving that these cells are susceptible to infection. Here you can see corneal epithelial cells. That's the clear skin that grows over the cornea, which is the clear shield in the front of your eye. And here you can see the Hong Kong virus and the Washington virus. And interestingly enough, the Washington virus showed an infection. You can see the red cells that were infected. The Hong Kong virus did not in this case. However, in other samples, we did see some infection by the Hong Kong virus. But there was some variability between donors, and there was some variability between the 2 strains. The Hong Kong strain behaved a little bit differently from the Washington strain. And then you can see here an additional sample of conjunctival cells. In this case, the Hong Kong cells, the Hong Kong virus actually caused quite a bit of infection, but we didn't see any infection with the Washington cell. So there's some difference between the viral strains, and there's quite a bit of difference between donors and also cell types. So there's quite a bit of variability, but ultimately every single cell type was susceptible to infection by basically both strains of the virus. And here's a summary of that. You see wherever there's a red plus sign, that's where we had a positive infection. And so we tested 6 different donors, and we tested the Washington strain and the Hong Kong strain, and you can see both strains were able to infect the various donors, and it was true for corneal epithelium, conjunctival epithelium, and limbal epithelium. So all these cell types really were susceptible to infection, although the rates of infection varied from tissue to tissue. The ND that you see here were cases where the cells didn't grow very well, and ultimately we didn't get a conclusive result whether it was infected or not. But you can see just from the presence of red plus marks in these 3 columns that all 3 cell types were able to be infected. We then looked at corneal endothelial cells, and these are cells that come from the back of the cornea, and these cells are, like I said before, the most commonly transplanted cells from the cornea. So increasingly, the most common treated disease for corneal transplantation is something called Foux Dystrophy. And in Foux Dystrophy, what happens is you have premature death and aging of these endothelial cells on the back of your cornea, and leads to swelling of your cornea, and that leads to blurry vision and pain on the surface of your eye. In order to fix this, you have to transplant new endothelial cells because the endothelial cells do not regenerate. So the only way to get new cells is to transplant them. What you're born with is all you ever get, and so you have to borrow donor cells if you want to get new ones. And so endothelial cells are the most common cell type that we transplant in corneal transplantation surgery, so we specifically wanted to look at these as well, even though it's on the inside of the eye and less susceptible to actual exposure of the virus and actual infection. Because this is the type of cells that we transplant, we wanted to make sure that these couldn't be infected as well. Here we did a study using endothelial cells that we had cultured out, and we checked with the Hong Kong virus and the Washington virus. We didn't see an infection initially, and we got kind of excited. We were like, oh, maybe the endothelial cells can't get infected. That means even though we have to screen out all these COVID positive donors, maybe we can still use the endothelial cells on the back of the corneas and use those for transplantation. Unfortunately, when we then went and did whole corneal infection studies, when we infected the whole cornea, we found that the virus could actually infect the endothelium. So in the cultures, we didn't see it, but when we infected whole corneas, the endothelium was infected. So what we did was we took what are called DSEC grafts, which are grafts that just involve the back side of the cornea and are used for transplantation of endothelial cells. We put those in viral culture. We put those in culture, and then we put viral cells live virus into the culture, and we checked and saw that actually in whole corneas we did have quite a bit of infection of the endothelial cells. Here you can see again the red cells that are stained with infection, and this is 2 different samples, and we ended up using the Hong Kong strain when we did this study because the Hong Kong strain was overall more virulent than the Washington strain. And then here you can see just a zoomed out picture. This is a donor cornea. This is something that would normally be transplanted into a patient's eye. And you can see these areas where the virus is slowly taking over the endothelial cells and growing out. And so this is not something you want to transplant into a patient's eye because it could definitely cause, I think, transmission of disease. And we don't know how well this cornea would do, even if the virus didn't cause a full-blown COVID-19 disease where they had lung disease and were in the ICU. It's likely that the presence of these viruses on the back of the cornea kind of eating away at these endothelial cells, it's likely that this graft would fail and not be a successful transplant in the end. And so more reason why we shouldn't be transplanting corneas from patients who are COVID-19 positive. And so again, we were basically the first group in the US and probably in the world to actually show with live infection, live viral studies that donor corneas and that the surface of the eye can be infected by a live virus, the live SARS-CoV-2 virus. And so we, again, were at the cutting edge. This data came out at the fall meeting of the American Association of Ophthalmology, and it was, like I said, the first real data showing that live infections can occur in corneal tissue. And so this was all driven by research that was funded by the Minnesota Lions Vision Foundation. It has really continued to drive what the CDC, FDA, and even the Eye Bank Association of America recommends as far as screening of donor tissue for transplantation. So yes, corneas can be infected. The infection rates vary based on the donor characteristics, the viral strain, and the tissue type. So in the end, what we really have to do is just rule out all COVID-19 positive donors to be safe. Now, we also wanted to look at one other thing, and that's whether the virus is very stable in the storage media that we put our donor corneas in. Because one of the things that we were hopeful for was that, well, the virus doesn't live more than a couple days on a surface like the tabletop or on your hands. So if the cornea is contaminated with SARS-CoV-2 virus and we put it in storage, can we just wait it out? Can you just put it in storage? Because the corneas are good for 2 weeks. Can you just wait 10 days and then the virus kind of dies off like it does if it's just on a tabletop? And then after it dies off, after 10 days, can we then transplant the cornea? So is there a way we can just quarantine it for a couple days and then it becomes safe to use? Well, in order to test that, we put live virus into the storage media that we use for storing our corneas for 14 days while we're waiting for transplantation. And what we found was actually that the virus is actually incredibly happy to just live in the storage media, and it does not die at all. It is just extremely happy. It's just a wonderful place for the virus to stay alive, and it does not decline significantly in terms of the viral titers over 14 days. So we normally store the corneas for 14 days, up to 14 days. Anytime within 14 days you can transplant the tissue. And we checked at day 1, day 2, day 4, day 7, all the way out to day 14, and we just never saw the virus titers really drop when we infected the storage media with the live virus. And then we tried it actually in a storage media container with actually a cornea in it, and again the live virus titers remained very, very stable and never really dropped over 14 days. And that means there's basically no sort of quarantine period that we can wait out the virus and sort of hope that contaminated tissue can kind of become decontaminated over time just as the virus dies off. Basically, within the window of time we have for transplanting the tissue, the virus is always going to be there. So again, we're left with the conclusion that it's really important to rule out COVID-positive donors because we don't have any other way to ensure that the tissue is safe. So in conclusion, corneal tissue does have the viral entry factors that allow the virus to enter and infect the surface of the eye and other tissues on the cornea. The cornea can be infected by live virus, and the variability between strains of the SARS-CoV-2 virus and donors and tissues isn't completely clear, but we do know that they can all be infected and that there is some variation there from patient to patient. And then we do know now that the SARS-CoV-2 virus can survive for up to 14 days in corneal storage media. So basically, if the tissue is contaminated, it's going to be no good, no matter how long we quarantine it for. And so based on these conclusions, the EBA has continued to make recommendations to screen out donor corneas that are from COVID-positive donors. And like I said, you know, we were basically the first in the nation, first in the world to publish live viral infection studies, information about corneal storage media infections, and we were still one of the leaders in publishing about viral entry factors. So we really were at the tip, and we continue to be sort of at the cutting edge of what is ongoing with COVID-19 research as it relates to eye banking and donor corneal tissue. And that's only really possible because of the support of the Minnesota Lines Vision Foundation. Now, future directions, obviously we don't want to rest on our laurels, and obviously COVID-19 continues to be a problem even with vaccinations. There are new mutations that are coming out. Europe has had, increasing outbreaks. Currently in 14 states in the US, there's increasing numbers of COVID-19. There's increasing numbers of COVID-19 infections. And so we're still looking at more research that we're trying to do that can help the eye banking community through the ongoing sort of pandemic. And what we are interested now in looking at is the epidemiology of viral mutations in donor eye tissue. Now, the CDC and everyone knows that the virus is mutating, and there's concerns that even when we're vaccinated, we may need to be vaccinated every year, every 2 years, that the COVID-19 virus may still be around, and it may be something like seasonal flu where you just have to keep getting vaccinated every year because it just keeps mutating and keeps evolving. And without continuously revaccinating, we might continue to have problems. There's questions about how long you're immune to the COVID-19 virus, even after you're vaccinated, how long that immunity stays, because if the immunity wanes over time, you may be susceptible again to a new strain of the virus. And so there's a lot of interest in studying what the mutations among the viruses what the virus is doing in terms of mutations around the country. And for Minnesota, we're particularly worried about the local mutations that are happening. So the best way to study this would be if we could get lung tissue from all the people who sort of have COVID-19, and that's not very easy to get lung tissue, to get biopsies of the lung, even from patients who have died and their bodies have been donated to research. Having someone who can go there in a timely manner and biopsy lung tissue is not easy. However, corneal tissue is a lot easier to obtain, and eye banks have infrastructure in place to procure research corneas all around the country, and we have a number of eye banks who have already started to collect corneas from patients who had COVID-19 because they know we're interested in the study, and they've sort of mobilized already in the anticipation that we are going to be doing this study and are looking to send infected donor tissue to us so we can evaluate some of the mutation patterns around the country. Once we get these corneas, what we have is a virologist here at the University of Minnesota who has a very cool technology that allows him to rapidly do full genome sequencing of the virus. And so within a couple hours, he can sequence the entire genomes of an entire batch of tissue. So if we give him like 30 corneas, he can sequence them all within a couple hours and tell you exactly what the genomes of all the different viruses that are infecting these tissues are. And you can actually compare the different mutations for every single piece of RNA in that virus. And so it is really cool technology, and it will allow us to really screen and find all the different mutations happening around the country from the donor tissue that we receive, and also locally from the donor tissue that we're getting from COVID-19 positive donors. And so using this, we'll be able to sort of get a sense of how quickly things are mutating, where the mutations are happening, where the mutations are happening into more virulent strands and where they're happening into more indolent and less dangerous strains. And this, of course, is something that's very interesting for the CDC, and we hope to use this data to sort of further fund a larger study that will be funded by the CDC. So these things are happening right now at the eye bank, again, with the support of the Minnesota Lines Vision Foundation. It wouldn't really be possible without any of their support. I, of course, am not a virologist, and none of this would be possible without all the support we get from the virologists at the University of Minnesota and other people at the eye bank. Dr. Qing Yuan is the research director at the eye bank, has been instrumental in driving this research forward, and I think he has sort of established himself in the eye banking community as an expert right now in cornea COVID-19 infections. Maxim Shiram, Declan Schroeder, and Dr. Krishna, they are the virologists who are working with us, and it is through their efforts and access to the Biosafety Lab Level 3 at the University of Minnesota through their connections in the virology department that we were able to do the studies we were able to do. And then Heidi is the manager of the core histology lab that we have at the University of Minnesota that provides services for the Department of Ophthalmology. And she has been instrumental in doing a lot of these stains that identify the infection of the virus, and all those pretty pictures you saw with the red stained cells were all done by Heidi. And so again, this is acknowledgement and special thanks to my entire team that has helped with this. Special thanks to the Minnesota Lions Vision Foundation for their funding and support. Special thanks to Lions Gift of Sight for supporting this research by providing donor tissue. And then we also had additional grant funding from the Eye Bank Association of America for this, and so I want to thank them as well. So that's it, and if you have any questions, I know this is a virtual conference, so you probably can't directly send them to me right now, but if you send them to me by email, I'll be sure to answer them for you. Okay, thank you.